Hello everybody, I'm Piero Carninci, and I would like to tell what I am doing in science. I was always interested in the function of the genome, but I realized since the time I was a student that it would be a very difficult task to analyze the function of the genome without developing new technologies. I was very interested in developing technology that can help us to make breakthroughs, and I moved to RIKEN in 1995 to develop a method to capture the genes as full-length cDNAs, so essentially capturing the whole coding sequences that are in messenger RNAs. I developed a technology, and this was used very broadly to understand the structure of the mouse genes and later the human genes. And this led to the construction of the Phantom project. In the Phantom project, we also identified new type of genes, genes that do not encode for proteins, so they are the non-coding RNAs, or very often they are also called regulatory RNAs or structural RNAs. And over the years, it came out that those non-coding RNA are actually a new type of genes. In developing other technologies to understand the genome function, I have also developed a second method called CAGE, or Cap Analysis Gene Expression, that essentially help us to measure transcription of genes and at the same time to measure the regions that regulate gene expression that are called promoters or enhancers. And using CAGE, we have been continuing the Phantom project in the Phantom 4 and Phantom 5 to create a very comprehensive map of regulatory elements. Now in the Phantom 6 at RIKEN, we are going back to the function of non-coding RNA, and we are testing the function of many of them. And non-coding RNAs are very important because we know, at least for some cases, that they can regulate the output of the genome. And in the future, we want to use the non-coding RNAs to really change the activity of the genome in specific points to address, to change the destiny, so to essentially cure a disease. And in the next effort, in collaboration actually with the Human Technopole, we will focus on the regulatory element and the non-coding RNA function and how they interact in the human genome, and in particularly across various populations, and in particularly also italic population. And we will continue to collaborate with the RIKEN Institute, because in genomics, the more data we compare, the better it is for the final outcome. I'm also interested in managing research institute, like the Division of Genomic Medicine here at RIKEN and the Center for Genomics at Human Technopole. We're aiming at creating environment that is very positive for the education of a new class, for the education of young scientists, and also to create one environment that is multinational and gives opportunity for the very large diversity of all the members that are involved in research. Thank you for your attention.